Okay, good morning. It is my pleasure to start the session, the first session of the day on stem cell technologies. And our first presenter is Professor Giuseppe Testa from University of Milan, who will speak about brain organoids-based neurodevelopmental disease modeling at a high resolution. So, Giuseppe, the floor is yours.
Thank you and good morning, everybody. I'm really sorry that I couldn't be there in person. It's great that normal scientific life is resuming, so I hope that we will have a chance to meet in person next time, which, as I will say towards the end, is going to be very soon. So, without further ado, let me go right to the topic of today, which I thought to structure in a way that would actually allow you to get a glimpse into what it means to aim at modeling human diseases today, especially brain diseases, using organoids. And just as a bit of introduction, in the lab, we study corticogenesis and its disruption, both when it is caused by genetic lesions, but also by environmental exposures. And over the last couple of years, we have become increasingly interested in the intersection of how neurodevelopmental disorders, which can be modeled through brain organoids, actually intersect the very recent path of our evolution and what distinguishes us as anatomically modern humans from our closest ancestors like the Neanderthals and the Denisovans. So this was just to give you a glimpse of the field we work in. And let me start with this quote, which will seem sort of old-fashioned to you, and which I think is important to capture where we stand in terms of biomedical modeling. This is the end of the 18th century, and Sydenham writes, in order to know the truth of the pathological fact, the doctor must abstract the patient, in the sense that he who describes a disease must take care to distinguish the symptoms that accompany it and which are proper to it from those that are only accidental and fortuitous, such as those that depend on the temperament and the age of the patient. So this is a quote which then served Michel Foucault to introduce in his seminal book, The Birth of the Clinic, the idea that paradoxically, in relation to that which he's suffering from, the patient is only an external fact. The medical reading must take him into account only to place him in parentheses. What did he mean by that? He meant that certainly throughout the 19th century, and I would say through much of the 20th, and in a sense still today, our approach to medicine has been, and still to a large extent is, an approach to diseases, to categories which are then embedded in individual patients, but always, in a sense, being careful to do exactly the kind of exercise that Foucault was reminding you, namely to abstract the patient. And that's why I've drawn here a parenthesis, and the fact that the parenthesis can now be actually broken open because we can put the patient in a dish in increasingly sophisticated models, which largely revolve nowadays around organoids. And so, in fact, it's not only that we can do it, but it's that the very idea of current biomedicine, in this wonderful review by Eric Topol and Dalit Torkamani, is that of a high definition intersection of the very features of specific patients. You see highlighted in yellow below from this cell review a number of terms which make up the discourse around precision medicine or high definition medicine. And you see that the idea is actually not to study a disease in abstraction, but that specific disease with this very ambitious and to a certain extent still very radical and very challenging goal, which is, as you read here, to compare you to you and to people like you and not to broad population norms. So really the idea that far from putting the patient in parentheses and neglecting those idiosyncratic personal things, you remember this sort of old-fashioned language, the temperament of the patient and so forth, so we will now call it in different ways, but the point is that the epistemology of contemporary medicine is precisely the opposite, as the one that we are coming from and that is still to a large extent being studied in schools. So since this is a school, I thought that it was actually important to start by enabling your focus on this new horizon. And now, how do we do that? So this is another great scholar, and I like to cite him also because Georges Canguilhem wrote what then became this foundational book for the field, which is still very actual today, The Normal and the Pathological. He wrote it as a PhD thesis. So I also like to cite this as a sort of a wish and a stimulus for you to actually aim at writing PhD theses that then can stand in history, hopefully with the same type of legacy. And in this book, he reminds us that to act, it is necessary at least to localize. Now, this may seem obvious, but it's instead depicting one of the fundamental issues of current human neurobiology. Namely that over the last 2 decades, we have learned a great deal about the genetic and also the environmental causes of neuropsychiatric disorders. And yet those arrows are still broken in the sense that we don't really have a good idea of the scale at which those causes actually manifest themselves in a way that is meaningful for observing the phenomena and even more so to act upon it. So to clarify a bit more what I mean, this is from a tweet by Kevin Mitchell, who actually articulates this in greater depth in his wonderful book, Innate. And here he was making a point between a cancer cell and, for example, an autistic or a schizophrenic neuron. And his point is expanded here. And this relates directly to what I was telling you about Georges Canguilhem and a necessity to localize. Namely, what he argues is that, especially for psychiatric conditions, they may have genetic origins, but they do not have necessarily proximal genetic mechanisms. And what he means by that is that these diseases reflect, the brain diseases, reflect altered states of distributed brain circuits and systems. Genetic variation may cause such a state to emerge, and we know that it does very often. But there may be nothing in the molecular functions of the affected genes that specifically relates to that state in any acute or ongoing fashion. So you see here the fundamental problem of scale, which is a problem of time. He's basically telling you that by the time that you see the patient, even if you know the genetic or the environmental cause, there may be nothing in that moment that relates that cause to what you're observing in terms of phenotype in any acute or ongoing fashion. And he's telling you that the scale at which these diseases manifest themselves, the level at which they manifest in terms of phenotypes and endophenotypes, may actually be very far removed from the original causes. So this is a problem. However, it's a problem that also gives us a tremendous opportunity for our generation, for your generation, that is actually doing science in this time in history, because of what I've called here the digitally scalar biology, namely the fact that if you think about it, the core of the contemporary life sciences is about encompassing an increasing range of biological objects and functions in a digital format. And once you have done this operation, you can actually intervene into these digitized objects and functions precisely through the digital codes. And this means something very practical, that if you think, and you see it below here with the green stripe, so if you think of even a drafty list of the levels of biological organization, I've listed here from the genome to an ecosystem, you see that all of them, to a large extent, have been digitized or are in the process of being digitized. And once you reduce them at the end to a problem of sequencing, you have already seen yesterday a couple of very relevant examples on this, then you can think of actually aligning these layers because you have de facto reduced them to the same matrix. So in fact, I am sort of putting here to exemplify this point, this half-joking tweet from Genentech a couple of years ago, saying that it would take 78 million tweets to actually convert the human genome to the new measure unit of a tweet. which is half a joke, but tells you that obviously we are embedded in a social life that is so pervasively digitized that we can actually remeasure our genome in terms of these, in terms of a tweet. But more seriously, that you see on the right, namely the very intense effort by a number of actors worldwide to embark in so-called digital phenotype, namely to take stock of the fact, as you read from the title, that human neuroscience, which of course studies human cognition and behavior, has become so pervasively digitized, the very fact that this lecture is being recorded, that we are actually increasingly digitized in the manifestation of our brain activity, that of course this can be captured also in digital format in a way that can be overlaid with all the other layers. So the implications of these are profound, because when it comes to modeling, and when it comes to modeling those genetic and environmental causes that I alluded to before, we have a major problem that you see depicted here, namely the fact that the knowledge about causation of these disorders is marching on at skyrocketing speed, but experimental neurobiology, namely the making sense of the scale, the drawing those arrows and filling in those still very significant gaps, is lagging behind. And is lagging behind because it requires functional experiments which still proceed at a pace that is incommensurable to the discovery of causes. And so this brings me to the second problem, namely that yes, it is true that the digitally scalar biology, namely the idea that we can combine together different scales of biological function because we have understood them and framed them as digital, find a major challenge in the problem that for each of those levels, and so, even if you think about sequencing each of those levels or at any way digitizing it, what really makes the difference, if you want to succeed, is the very scale, now in the second meaning of the word, namely the amplitude, the size of the sheer effort. And this is summarized here in one specific case, which is particularly close to what we study in the lab, namely autism spectrum disorder. But the very same thing could be said for a vast majority of brain diseases, and has been captured very nicely by Stephen Hyman when he talked about the daunting polygenicity of mental illness and the need to make a new map. So much so that he explicitly talked about the need to, and the situation of that we're in, and the need to map a terra incognita. And the reason you see it below is that over the last 10, 12 years, we have fragmented disease categories into a multiplicity of rare diseases, and this is for the rare variants of high penetrance. So we have broken categories such as autism and several others into a huge collection of rare diseases on the one hand. But on the other hand, we have also recognized the extreme polygenicity of a lot of these conditions. And this means that, again, we are fragmenting disease categories, but this time not into rare diseases, each of them caused by a variant of high penetrance, but rather in so-called genetic loadings for a multiplicity of individual risk alleles. And so if we need to make a new map, let's pause for a moment to reflect what we should expect from a map. And we are talking about brain diseases, and we would like these maps, as I've written below, to actually be actionable. And if they need to be actionable, they almost invariably need to be mechanistically validated. And hence they must have single-cell resolution. And importantly, for all the reasons that we said before, namely for this epistemology of high-definition medicine, we want to be able to do this at a scale that is relevant for the patients. And so here I put on the screen for you this famous short story by Borges, which doesn't cease to capture my imagination. It's a sort of a dystopian in which cartographers become so good at making maps that the exercise becomes completely pointless, because the map is one-to-one linear with the territory that it is supposed to represent. And of course, this makes a map useless. So in order to be useful, a map needs instead to do what is depicted on the left, namely to invest, to give us a compass, to move in the territory. In the territory, in these analogies, the territory of real life, the disease, in how they unfold for the brain, for the, in a patient's history throughout their lifetime. And once this map allows you to make some mechanistic predictions, hopefully invest the territory again with these predictions, and by analyzing what happens once you invest the territory, namely once you go into the real life and try to change the course of the disorders with what you have inferred from the map, go then back to the map and try to improve it in a virtuous iterative process. And we now have the hope to be able to do it, and it's a concrete hope, because in many ways, as I've argued with my postdoc Niccolò Caporale in this chapter, we can actually in many ways replay Vesalius. The analogy here is with Vesalius, who, as you know, in the Renaissance gave us a completely new resolution and definition of human anatomy. Through accuracy of drawings of human anatomy that was unprecedented, and which I think stands as a relevant counterpoint to where we are today, because of course now also we are in an age of unprecedented resolution. Think about all that we can measure in single cells, and so how deep our molecular gaze has become. But of course, crucially, and this is the point of this quote, we have added the temporal dimension, because of course Vesalius could do nothing but observing the bodies of humans, but these were dead bodies. And here instead now we can extract from individual bodies through cell reprogramming, and these times therefore the bodies are living rather than dead, not simply organized flesh, but organized flesh captured in its making. And that's the whole point of organoids, and especially brain organoids modeling, namely the possibility to recapitulate development in vitro and to do so at patient-relevant scale. And so for me, human brain organoids are really best conceptualized as what I've termed here the developmental phenotyping conduits, namely tools that allow us to probe development in vitro and thereby make strides into a number of endophenotypes which can connect those different scales that I mentioned before. And in the second part and final part of the talk, we will be seeing also about how we are attempting to solve the second problem of digitally scaled biology, namely the goal of actually doing this at a scale, namely at a size that is relevant for the effort, given what we said about the polygenicity of these conditions. So to illustrate this point, I always like to actually juxtapose these two Dutch masters to make a point about what we expect from a modeling effort of the brain and why organoids are so relevant. So on the left, you see a Mondrian, and of course, it is easy for us to imagine that the painting would have ended up just the same, regardless of where Mondrian would have started to paint it, namely from the upper left corner, from bottom right corner, and so forth. Whereas the painting on the right, the Kooning, is what I like to call a thoroughly developmental painting. Namely, it's impossible to even contemplate that you would have had the same result had the Kooning chosen to have a white brush instead of a red one, or whether the blue stroke would have preceded the yellow stroke or followed it, and so forth. In other words, the one on the right is a thoroughly developmental painting, and not by chance this comes on an analogy originally developed by Andrew Pickering in the context of the philosophy of science, which I then applied to the magnificent work and legacy of Annette Karminov-Smith, who has been championing the so-called neuroconstructivist approach to brain diseases, which is an approach that takes development as the fundamental aspect that we need to chart in order to understand these diseases. And so once we are able to do it, we still, however, face a major issue, namely that if organoids are these developmental phenotyping conduits. They act as experimental models that you see in the middle. Then they can serve to actually validate the relationship, as I alluded to before, between the map and the territory. In other words, currently our maps, and we have started seeing them yesterday, and you will see many more today and in the days to come, also of course at the ICCR conference, the maps are exploded. We can probing, increasing numbers of cells for a number of features that you see listed below. All of them are becoming increasingly amenable to increasing single cell resolution. And yet for each given disease, we really do not know how many layers we will have to profile, which ones, when, how often. And especially we don't know how much of these will actually be relevant to tackle either of those aspects on the right, namely what we are interested in, which is changing the territory, the real life manifestations of these disorders, all the way from their prevention to the sustainability of a healthcare system that faces brain disease as one of its major societal burdens. And so in order to do this, together with several other colleagues, including a number of Israeli friends through the years, so we have been championing the vision of so-called intersecting medicine, which in my view is a way to take the intuition of Foucault and actualize it, namely to break open those brackets and actualize the patient in a dish approach through technology that makes the effort feasible at the patient relevant scale. So here you see a depiction of this major European effort, which is still continuing in various forms. It was originally conceived within a FET flagship application, namely the idea that for each of the main disease domains that afflict our society, including, of course, the neurological and the neuropsychiatric diseases I am focusing on, you can think of disease as a rupture of the cell homeostasis of tissues in time. And so what we would really like to do using single cell resolution and organoid models directly sourced from patients is to measure finally this angle of interception, namely the earliest possible deviation over time in which a given tissue, in this case the neural tissue, starts deviating from its original track. And so to do this, of course, it's crucial to understand how much these experimental systems that I've been discussing, namely brain organoids, can reveal and recapitulate human brain development, in our case, especially for the cortex. And so this is a very large effort that has been going on in the lab for quite some time, and which builds on a number of efforts over the years in which we aimed at developing or refining tools that were specifically geared for the challenge of human disease model, a challenge that became very evident in when we undertook this work benchmarking the two largest resources of induced pluripotent stem cells in the world, one in the UK, one in the US, to actually see whether we could define empirically grounded recommendations for the optimal the design of IPS-based modeling studies. And what we found is that, of course, even just at the level of the transcriptome, what was happening is that with the current design in terms of number of lines, number of clones, and the kind of statistics that was being used, a lot of times the studies were actually ending up detecting as disease-relevant differences things that were actually just the result of the human genetic heterogeneity and variability. It is just to say that when you study humans and when you want to be deep about human disease modeling, you must take, obviously, human diversity into account. And so we ended up with making some empirically grounded recommendations, which then gained wide traction in the field, and which was also the inspiration for actually doing a very similar exercise for brain organoids, namely benchmarking the extent to which brain organoids recapitulate fetal corticogenesis, combining both a from-scratch meta-analysis of transcriptomic data sets from different brain organoid protocols. You see them here, some of the main ones, but also doing in-house cortical organoids dataset and generating fresh new data to measure experimental variability across both individual organoids, multiple genetic backgrounds, and multiple batches of differentiation, all the way from the start to a stage that approaches late gestation or perinatal cortical development. Then we integrated all of these data sets, including data sets coming from primary fetal tissue. And I'm going to show today the main results which are relevant for the topic of the lecture as an introduction to the scope of brain disease modeling through organoids. So first of all, we applied the technology called WGCNA on the brainspan, which is this catalog of transcriptome of the developing human fetal brain, so of what happens in vivo, to uncover from scratch transcriptional patterns that are modulated during the development of the fetal cortex. You see below some of the main ones, some of which are positively correlated with the sample stage, because of course brainspan has samples throughout the stages of human fetal development, and some that are negatively correlated. And then we, of course, asked how these modules, how these networks and how their pattern of expression in vivo matched to the available in vitro models, so to the brain organoids. And you see here that there is a preservation of the pattern of and of the ontologies of these gene networks pretty much across the organoid systems that we looked at, which are examples of both pattern organoids, such as the cortical organoids, and non-pattern, such as the whole brain organoids, the forebrain organoids, and the telencephalic organoids. And you see, however, that we also identified a couple of modules that I call here, which we termed biphasic, in the sense that they actually have this interesting pattern of being first down-regulated and then being up-regulated again during the full span of fetal corticogenesis. And these are interestingly enriched in extracellular matrix and morphogenesis regulation. So we asked what happened with these two modules that have this distinct pattern. And as you see, we actually find that these modules are more closely recapitulated by one type of brain organoid protocols, the cortical organoids, which are patterned at the examined time points, rather than other systems, which, as you see, there is, in fact, a completely different pattern in terms of these biphasic modules. So this was interesting for us and actually pushed us to do a cross-validation of these strategy. Namely, we asked, here we started by mapping these modules on in vivo, on brainspan, and then charting their accuracy in vitro. Can we do the opposite, and can we learn something from the opposite? Namely, can we start with an interrogation of cortical organoids and derive in cortical organoids the gene networks doing exactly the same approach, and see to which extent we find an overlap with the networks and the modules that we identified in brainspan. And we do find a significant overlaps, especially for the modules related to neuronal maturation, to cell cycle, and to the extracellular matrix. And importantly, when we now have the modules that they arrived in cortical organoids, you see that also when we start from scratch in the in vitro models, we actually do find these two modules that have this characteristic biphasic pattern. Unsurprisingly, this pattern is reverberated in the in vivo data set, but it is actually not maintained in other protocols. So this was, on the one hand, a validation of the fact that starting from completely independent data sets in vivo and in vitro, you can actually reconstruct a core of regulated pattern, and that this illuminates how different protocols are actually quite distinct in the way in which they recapitulate, especially developmental time. So this is summarized here. in which you basically see a heat map that aligns these different organoid protocols to the weeks of gestation as captured in the transcriptomic analysis of the brain span. And these meta-analysis reveals some differences in the recapitulation of timing, which is important because remember what we said about the need to actually choose the right model to make the right kind of map for a given purpose. And so these different protocols are likely going to be differentially useful depending on what the goal is, because the cortical brain organoids increasingly correlate with post-conception week along the differentiation. We confirmed what had already been observed, namely that there is a rather fast evolution of the system, a rather fast kinetics of the system initially, and then there is a slower phase after day 100. And importantly, for the benchmarking approach that I was mentioning previously, you see that each of these columns is a specific line, so a different genetic background, and you see the very high reproducibility of these protocols between lines and replicates. Some of the other protocols instead have two interesting features. First of all, that, for example, the cerebral organoids at day 40 and 60 correlate with the post-conception weeks, for which similar levels of correlation for the other protocols are detected only at day 100. And even a sort of greater acceleration is observed also for the other protocols, which basically means that in vitro time is differentially accelerated by different protocols vis-a-vis the in vivo gold standard. And importantly, some of these protocols, especially this one that you see here, appear to be much less tolerant to inter-individual variability, because you see how individual lines behave differently at the same stage. So these are all considerations that you want to take into account, and that the community needs to take into account when actually choosing specific models to recapitulate both the physiology of development and even more its pathological alterations. So this is just to give you a snapshot in a very nice review by Giorgia Quadrato of the investigations that have been done with single-cell omics of human brain organoids, including our own from 2.5 years ago, in which we interfered with GSK3 and by inhibiting these fundamental regulators, we also provided the first example of the use of single-cell transcriptomics in brain organoids to actually dissect a phenotype. And in that case, the phenotype was that by impairing GSK3, we actually found that there was an impairment in a very important neurogenic path of the human brain, namely the generation of autoradial glia, which is a hallmark population that underlies the expansion of our cortex. So we are currently engaged in expanding the repertoire of region-specific organoids, and you see here the people in the lab who are spearheading this work, especially focusing on further on the cortex and including also the thalamus and the cerebellum. Here you see just examples of now of how also these regions can become amenable to brain organoid modeling. And this is just an example in which you have started to operate these two movies, in which you are seeing here a model of interneuron migration when we actually fuse in assembloid, as originally developed by Sergio Pasca, ventral and dorsal for brain organoids. And you see that one of the key features that is recapitulated in this more complex system is that the direction of migration is one way, namely from the ventral to the dorsal, and is instead different when we fuse the thalamus and the cortex organoids, in which we actually find that there are projections in both ways, as we expect. And what we are currently doing is to actually fusing all 4 of these organoids to start building systems that allow actually to enable the initial platform onto which prospectively model cerebello-tatalano cortical circuits, which are involved in a number of key functions, especially relevant for us. They are involved in associative functions that have become very relevant in the pathogenesis of autism and intellectual disability syndromes. So let me then go back for the last couple of minutes of the talk to this initial point that I labeled, namely the fact that the complexity of the genetics of these disorders, let alone the complexity of the environmental causation that I will talk more about during my talk at SSCR, so that you can think of this lecture as also preparing you for some of the aspects that we will cover in the talk. And you remember here the point was about the complexity of these disorders to the extent that we are in a terra incognita. And what we have been doing in the lab is both to focus on those variants of high penetrance, rare variants of high penetrance, as exemplified here, but also increasingly moving towards the possibility to study variants of low penetrance that together are combined into these loadings of differential vulnerability and resilience for people who will then go on to develop this disorder. Now, when we think of genetic and environmental causation, we need to include a third aspect, which I've mentioned already throughout the talk, but now I mention it in a slightly different flavor, namely to think of the development as the third fundamental axis of variance. In other words, a lot of the neuropsychiatric disease variance is due to genetic causes, some of it to environmental causes, but then there is a third element, which is development itself, namely the fact that the development is an inherently stochastic process, has an inherent degree of stochasticity embedded in how it unfolds, and these may well be the reason for why, for example, certain variants do not end up in a phenotype. And of course, this has been thought and contemplated in the field for a long time. What, however, has not really been possible until very recently is how to actually put it on a secure quantitative footing. And so, since this is a school, I'd like to conceptualize a couple of last points, and this is really an inspiration that came from this very nice review in the development by Denisha Badon and Madeleine Lancaster, exploring landscapes of brain morphogenesis with organoids. This was a review of a meeting that was held in which the notion of the morphospace was, in a sense, resurrected. The morphospace was a concept originally developed in evolutionary biology and paleontology by Mitterrocker and Hutteger about 7 years ago. And James Briscoe put forward this concept again during this meeting, thinking about discrepancies between in vivo and in vitro. I've shown you also today some of our work that actually looked at the discrepancies between in vitro and in vivo in terms of acceleration of corticogenesis time. And so here the discussion at the meeting was whether the discrepancies in the development could actually be informative to understand the realm of possibilities of a given group of cells. In other words, even when organoids in vitro seem to do things that are different from their in vivo equivalent, they can still be relevant because of what you read here, namely, it illustrates the subset of realized forms. So the topic is the Themorphospace, illustrate the subset of realized forms of a structure relative to all possible forms, providing insight into the constraints that operate to restrict the evolution of a species or a clade. This was the original meaning from paleontology. In the same way, organoids allow the exploration of the space of possible forms, of which the one realized in an embryo is what the embryonic constraints determine. Anomalies in vitro, so these discrepancies between in vitro and in vivo, may therefore inform us on the plasticity and the developmental potential of cellular assembly by untethering, so by dissociating the developing tissues from the systemic in vivo constraints imposed by millions of years of evolution. So this was an interesting way to say, well, what can we learn from these in vitro developing systems, even when they do not match closely or one-to-one the in vivo counterpart. So here I propose that we are now ready for a number of the developments to make a further leap, namely to apply Morphospace to biomedicine. So to complete the transition from paleontology to developmental biology, now to biomedicine. And here I wish to redefine Morphospace in this project as an exploration of the space of possible developmental forms for a given human genome, to quantify the possible source of developmental variance for any given set of genetic and environmental variables. In other words, if we know from the way that we have measured it over populations, and of course from what we have been knowing for decades from the twin studies, that genetic and environments are two major sources of variation depending on the various diseases, with a clear prevalence of genetics as far as we know today, but that development plays also a critical role as a source of variance. The problem was that until recently, the development could not really be recapitulated in humans, and each of us developed, of course, only once. So this was impossible to measure. But now that we can replay development in vitro, at least to a significant extent, this gives us also the way to measure the possible developmental forms for a given human genome, namely for a given setting of those genetic loadings, and possibly also for those environmental loadings. And so how do we do this? Well, in order to do this, we can again think of the analogy that I drew before. Here again, the Koenig and the idea that with IPS reprogramming, so in the last really 15 to 16 years, we have learned how to go back and how to go forward again, especially now that we have the possibility to move forward in 3 dimensions with increased accuracy, as I've shown you today. And of course, we can actually now measure what I told you was going to be very difficult by definition. Namely, I told you that it was impossible to even think of the Koenig that would have painted the same thing in exactly the same way had the paintbrush followed a different course. And now, however, we are in the position in which we can measure exactly how broad or how narrow that space of the Koenigian possibilities, so to say, for a given human genome, because we can move forward in developmental time in vitro as many times as we need in order to actually measure the morphospace of a given human genome. But to do this, of course, we need, first of all, to move from bespoke tailoring to pret-à-porter. That's why I am putting here on the screen Yves Saint Laurent with the famous Rive Gauche collection that marked this transition in the history of fashion, precisely because we cannot do this kind, we cannot engage this kind of scale if we do not have the possibility to move from an artisanal process of brain organoids production to a process that generates pret-à-porter and industrial-like methods. And so that's what we are very heavily working on at Human Technopole in the Center for Neurogeogenomics, in which we have set up a facility for the automated reprogramming, editing, and longitudinal brain organoidogenesis to serve our community and, of course, the extended communities of scientists through a number of collaborations that we have already started. And the idea is really that these should move us towards the ability to actually use entire cohorts and in prospective populations onto which to do the type of modeling that I showed you today. And this is done in a very close interaction with the heads of this facility, Giovanni Faga, and the head of the image analysis facility, in a workflow that, as you see below, automates all the manual tasks via robotics and microfluidics, all the tasks involved in reprogramming, editing, and brain organoidogenesis, and at the same time, in parallel, automatize the complex decisions by artificial intelligence, the convolution, namely the automation of the selection of organoids, and aims at increased throughput through adaptive fast imaging and online imaging restoration, finally aiming at what we call the unsupervised phenotypic discovery, namely from these longitudinal pipelines of brain organoids growing in vitro and observed through time, the possibility of deconvoluting new endophenotypes that can then be matched to the clinical history of patients. And so this is then one of the cornerstones of the way in which at Human Technopole we are starting to do human experimental neurobiology at scale with a circuitry that brings you from those densely phenotyped cohorts. So the way in which the clinical focus that I started with today, thinking about the relationship with the medical gaze at the time of CD-RAM and Allertime, and linking these phenotyped, deeply phenotyped cohorts through two molecular endophenotypes to this level of scaling up of organoid editing technology, and of course, including in vivo validation. We are currently in a very hot phase of recruiting for a number of positions. So in case some of you are interested, they can just drop me a line or even text me directly on Twitter. This is a Human Technopole where this research is taking place and where the center is shaping up. And last but not least, see you in Milan, because as I'm sure you have heard already, we will have finally also in person our Episystem closing conference on stem cell epigenetics in July from 4th to 6th. And this will bring together the fantastic community of the Episystem network and many of the great scholars and friends that are also teaching at this school. So I look forward to seeing as many of you as possible in Milan in a couple of months. And with this, I finish and I stop sharing.
Giuseppe, thank you for a really thought-provoking talk. Now questions from the audience.
Can you elaborate on the technology?
Can you speak up?
The technology for creating the organoids, besides that it is automated, what is the basis? Is it the gelatin? What's the base of the organoid?
So I'll repeat the question. Can you elaborate on the method that you generate the organoids in an automated manner?
Yes, I can elaborate to the extent that we have been spending about a year and a half to 2 years to actually translate the various bits of the workflows of a number of protocols, starting from the pattern protocols for the reasons of reproducibility and adherence to the developmental time that I showed today. And we have been actually transforming these individual bits of the workflow into processes that actually could be automatized. And then in selecting the automation platforms and the software solution that were best attuned to actually combine all of these workflows together. This is largely the work of Niccolò Caporale, Emanuele Villa, and Giovanni Fagan. And we are very now hopeful that these will actually be able to be translated into a new edge for the field. So this is what I can say at the moment. I'm not hearing very well.
So the question is, I'll repeat the question. So the question is, what are the beads made of?
I think there was a misunderstanding. I said bits in terms of, parts. So to actually divide this very complex workflow into small parts, each of them that could be automatized and then put together. So it's not beads, it's bits.
Yeah, bits.
I hope this clarifies the issue.
Maybe that's a better offline question. Other questions? Yes. Yeah, Oren.
So Giuseppe, thank you, very interesting. So you said that development is stochastic, but it's also very robust because we all end up with the same brain more or less. And yet, some protocols show the differences between cell lines and some don't. Do you think it may reflect on the level of stochasticity in our brain, that some regions are more prone to stochasticity than others?
So thanks, Aaron. I think that this is a very interesting thought. And I completely agree with you that there is, of course, as I said, an inherent element of stochasticity within, however, a process that remains very robust and resilient. And I think that the challenge is precisely to actually measure now that we finally can, hopefully, what is exactly the degree of stochasticity. And you're asking, I think, a very thought-provoking question about different brain regions, possibly as a result, I would say, of differential evolutionary constraints, have evolved to allow different buffers, sort of to accommodate different degrees of stochasticity and vice versa, different degrees of robustness. That's, I think, a very nice thought that with the tools at hand, we should be able to answer. In terms of the other part of your question about the protocols, part of this, of course, depends on how heavily patterning the protocols are, because it's clear that the more you pattern, the more you, in a sense, constrain the system. So you are almost necessarily, I would say, bound to actually constrain also the variability among backgrounds. So in comparing protocols, of course, this factor must be taken into account. But I think that once we have, in a sense, regionalized protocols that are comparable at the level of constraint, we can then explore whether inherently different parts of the brain have evolved different ambits of stochasticity. I think it would be sensationally interesting to do it.
Thank you.
Okay. We have time for one last question. Yeah.
So, I believe in organoids, and I just want to say, I'm just playing the devil's advocate here, but you mentioned that you want to look at the developmental variance, aside from the genetic and the environmental cause. So that means that you should see, if I understood it correctly, you want to look for an anatomical proxy, the organoid, to give you a mark of the disease, preceding the disease or as a mark of the disease. Is that correct to say?
So there are two aspects here in the question. One is to actually use organoids in many systems, including the brain, which of course is one of the most difficult in this aspect, to actually capture initial deviations of the normal trajectories, which then seed the disease. And the other aspect is instead to actually measure the morphospace, meant as the range of options that given genome has at its disposal in the development. These are two aspects which are different, although obviously they interact.
But specifically, let's say with the developmental variance, and you see developmental perturbations from the normal trajectory, and that would be a correlation of the disease or a marker. This is what you're saying, correct?
Sure. In some cases, we expect. Sure. In some cases, we expect that an aberration in developmental trajectory, as we and others have shown, is actually a anticipation, or in fact, a cause of the endophenotypes and the further manifestations of the disease. Sure.
Okay. But so now this is the real question then. If that is the case, and that's, I guess, in many neurodevelopmental pathologies, that would be the case. But let's say the devil's advocate, let's say you look at neurodegeneration like Alzheimer's, where many times the anatomical correlates come quite a bit after the behavioral pathology, like the tau amyloid plaques. And even in certain cases with autism, now people are claiming the dendritic pruning and arborization defects might come many years after behavioral diagnosis. The gold standard for developmental disorders like autism would be the DSM-5 guidelines, which is rather subjective. How would you reconcile that, I guess? If the anatomical proxy, let's say an organoid, comes theoretically in a human patient after the actual disease pathology.
So, if I understand correctly, because I didn't hear very well, the question is whether the timing of certain anatomical correlates, which may well actually come after the initial clinical manifestations, how would one tackle that in organisms? Is that the question? Did I understand correctly?
Yeah, perfect.
Yeah. So, first of all, I find it very interesting both that we are in an age in which, for certain neurodegenerative diseases, think also of Huntington's, we are increasingly understanding that although the disease is framed as a late onset disease, actually its seed are apparently sown very early in the development, and vice versa, what you are saying, namely that there are certain anatomical hallmarks of the disease which may actually be very late consequences and may accrue much further than the disease onset. And I would argue that these systems can allow us precisely to do that, because we can move them in time, and we can, of course, try to actually see, and this, of course, is a major challenge, to which extent that there are aspects that relate to the onset of disease, and to which extent there are aspects which are actually only much further downstream. Now, obviously, this is, I think, precisely the challenge that I was alluding to at the beginning when I talked about scales and of the broken arrows, because the issue is how do we actually bridge all of these gaps, many of which have to do precisely with the timing difficulties that you're mentioning. And I think that by having the possibility to move back and forth in time, of course, matched to an equal developments in the clinic, in which, of course, we don't have obviously the way to move back in time, but we should have increasingly the way to actually have much more detailed histories of the symptoms over time, including through digital phenotyping, and find anchor points that actually match in vitro and in vivo. That's at least my hope at the moment.
Okay, thank you very much, Giuseppe. Let's thank you again. Thank you. And now I'd like to introduce Oren Ram from the Hebrew University. We'll speak on CloneSeek. רגע, הצידה, צדדים, תודה. Okay, CloneSeek, a highly sensitive single cell based platform for comprehensive characterization of 3D cultured cells.
Thank you very much. אני צריך לחבר את המחשב. כן, יש לי פה טלפון, אבל תגיד לי. יש כזה מערכת שקפים? הבנתי. אז רק רגע. Okay. Thank you for the organizer for the invitation. So our lab is located in the biological chemistry here in the Life Science Institute. And in the lab we are studying early differentiation. This is the main topic, and cancer. But in the process we're also developing new tools. Today I would like to talk, if time will permit, about two projects. First one is technology development. We call it CloneSeek. It's actually trying to overcome a limitation of the regular single-cell RNA-seq technology. And in the second part of the talk, I would like to talk about early differentiation, exit from pluripotency, and tackle also one of the major confounders of single-cell RNA-seq, which is cell cycle. Okay, so the first project is spearheaded by Shue, Dani, and Chen, PhD students in the lab. So currently, single-cell technology can be divided into two types of technologies. The first one is the low throughput, well-plate-based, which allow us usually to retrieve decent amount of RNA molecules, around 35,000 molecules. If we talk about the 3D digital counting, when we count the 3 end of the transcripts. But the limitation of the low throughput obviously, is the number of cells. So we have limitation is the statistical power to find rare cell population and so forth. Currently, most of the world are using the high throughput technologies, mainly drop-based microfluidics or sequel, nanowells, and also some kind of pull and split barcoding systems that allow us to profile many cells. However, those high throughput technologies which allow us to process many cells in the same experiments are limited in their sensitivity and coverage, and most of them can provide us around 5,000 to up to 10,000 transcript, RNA transcript per cell, and obviously this is a limitation. And the main limitation, so first of all, single-cell RNA-seq is a powerful technology, important technology that we can leverage and study many types of biological systems, but it's limited mainly to be able to recover and divide between cell types. So if we take a brain, you can easily find different cell types within the brain, neurons, glias, oligodendrocytes, and so forth, and mainly because those subpopulation or cell types are really divided by highly expressed genes. However, there is an elephant in the room, right? So if the only way to divide between two types of cellular states and not types is lowly or mid-level expressed genes, then we might have a problem, right? And if we look on PC9, non-small cell lung cancer adenocarcinoma, then most we can find using single-cell RNA-seq is cell cycle based separation. If we look on mouse embryonic stem cells, even not in ground state, after knocking out OCT4, so we might have some differences between the subpopulation, but they are not clear. Okay, so the main limitation of single-cell RNA-seq is the fact that we're actually looking on highly expressed genes. There is major confounders like cell cycle, transcriptional bursting, which is a stochastic effect of gene regulation, and a lot of noise. And we are limited. This is one of the limitations of the technology. So we decided to approach this limitation instead of trying to improve the technology itself, the technicalities, the RT, the cell extraction, and so forth. We decided to improve the amount of biological material that we can actually extract from a singular entity.
Is that a limitation in cell lines that you do single-cell RNA-seq, or in vivo tissues where you isolate?
We don't even know, because if you look here in these excitatory neurons, they are all the same. We don't know, right? It might be that these hide many types of cellular states, but the technology just not allow us to see that. Okay, so we are kind of blind to only genes that are highly expressed. If you are running 10x genomics and you can process, I don't know, 10,000s of cells in one experiment, you might overcome some of this limitation just by the large number. But again, if there are two genes that dictate a subpopulation, what is the chance that you'll find these two genes on the same cell? So it will really dictate a subpopulation. So you might just lose that in the process. So our approach is, instead of looking on single cells, we would like to look on single clones. Each clone is actually developed from a single cell. Obviously, there are limitations, theoretical limitation here. We need cells that can replicate and divide, and we need to ask questions about the homogeneity within the clone. So I'll touch that in a moment. But currently, on the technical level, we need to separate cells in drops, and then we use PEG dextran chemical. This is two components. The PEG comes with thiol, the dextran come with malamide groups, and when they mix together to form these drops, together with single cell, they form hydrogel. This hydrogel can then break down, not the hydrogel itself, from the microfluidics. We take out the surfactant, we take out the oil, we extract the hydrogel, and we culture them with regular media to form these clones. So this is one cell with GFP knocked in inside. PC9 cells, and after 9 days of culturing, we get this nice clone of around 30 cells. We need to optimize the condition of the hydrogel in order to be able to grow different type of cells. So currently we optimize it for the PC9 cell, for mouse embryonic stem cells. Here you see the OCT4 in clones of mouse embryonic stem cells. This is kind of a bird view of many clones together in hydrogels. And we can also optimize it for K562 and even for primary glioblastoma cells directly from a patient. This is the number of cells we have after 7 or 8 days for PC9 or 3-4 days for mouse embryonic stem cells. The nice thing with this hydrogel that we can mimic the microenvironment of the cell. So we can add RGD group, we can add metalloproteases, different gelatins, and so forth in order to promote the growing of different cell types. We can also play with the elasticity of the hydrogel and so forth. Another nice thing with this hydrogel is that it's biodegradable. We can add an enzyme that break it down. And this is important for the experiment I'll just show you. The diameter is between 50 to 150 micrometer currently, and we can support the size of clones in the range between 1 to 100 cells. Above that, the cells can go out of the hydrogel, and then we have a problem of mixing. So now, to the most important question. Are we wasting our time, right? Maybe if this is like two hypothetical single cells that reside within two different states that we would like to learn about, if this option 3 is the option that actually happens, then we're wasting our time, and most importantly, I'm wasting your time, because this is just looking on the average, right? It's just taking bulk cells and profile them and trying to compare between biological replicates. But if this one of two options is the correct one, then we have the chance to improve our sensitivity to capture subpopulations. Right here, just the clone, the very small clone, is composed from the same state as the mother cell, and here we have enrichment, at least enrichment of this state, so computationally we can resolve that. So how we tested? I can hear you?
Saying that also you don't want that you use to affect the cell. Then you have a new cell. Like the hydrogen.
So what you say is it might be it's like that, but this is a different color from that.
Yes, like affect it. Especially if you try to find the genes that express in a low point.
Okay, I think if I understood you correctly, what we did in order to understand that, we just did some bulk experiment and show that single cells and cells that were growing in the 3D condition are very similar to each other. So we don't really adding a different effect here. But
It's something technical just to understand. If we look for endpoint, for example, and you track this cell and culturing them in this environment, this is one thing. The other thing, the cell need interaction. So how you get over these things by this technology? This is very important.
yeah, so first of all, they have the interaction with the cells within the clone, and there is a diffusion of nutrient and reagents between the hydrogels. So all the hydrogel are actually growing together in the same culture, and they are totally talking with each other chemically.
If you isolate one cell,
Yeah.
you reculture it, which will affect the transcriptome and will affect the...
Okay, so we tested it, and at least for PC9 and for mouse embryonic stem cell, it doesn't seem like it was affecting it dramatically. There are some changes, I agree, and we will need to figure out that in the future, I guess. But currently, that's the best we can do. And by the way, I'll show you in several slides ahead that we actually have some advantages in growing the cells like that. Okay? So how we figure that out? What we did, we just produced a PC9 line of cells with genetic barcodes within them. Okay, so now we have some statistical problem that we need to grow the clones, and then we need to collect only few clones in order to have a statistical chance to actually capture two cells that originated from the same clone. But eventually we could figure that out, so we made the clones, and after 7 days we used the fact that these hydrogels are biodegradable. We break the gels, extracted single cells, and proceed to single-cell RNA-seq. What you see here is 3 biological replicates that actually showing, so for all the gray dots here are cells that we couldn't find a pair from the same clone. But for the color ones, we found at least one or more. And then we did just, we calculated the distance between the cells, both on the UMAP space, but also in Euclidean distance. And we find the same thing. So cells that originated from the same clone are always much more similar to each other compared to cells that were picked at random. And that means that at least in the worst case scenario, we are talking about option 2. But that motivated us to proceed and see what we have when we trying and studying the clone-to-clone variation. So how we do that? This is a clone, another clone. Obviously, we have different sizes of clone. This is part of the heterogeneity that built into the system. We are talking about cancer cells. Not all cells have the same capacity to replicate. And this transparent hydrogel is the hydrogel with the RT primers, the barcoded RT primers. And then we tested 3 parameters. The first one is that we don't have leakage in the system. We mixed mouse clones with human clones. And as you can see, beside one event of mixing, it might be that we have other events of mixing that coming from the same organism, like 2 clones of mouse, but we can't really see that. But overall, it seems pretty clean. So this is the number of humies aligned to a specific organisms. And the most important part, maybe, is the fact that we actually have a better coverage. So if from single cell we have around 5,000 to 10,000 transcripts per cell, for clones, we have around 100,000. And it makes sense because the clones are on average of like 10, 15 cells. Another analysis we did is to see what is the coverage, the sensitivity of the system, and comparing single cells to clones. And again, single cells are cells that have been grown as clones and then separated into single cells. And they provide a very average quality of single cell assays. And when we look on one cell or one clone, we cover around 50% of the genes. But when we move the threshold and look how many of these 100 cells or clone are shared the coverage of the same genes, then we see that when we look on 100 cells, only less than 10% of the genes are being covered. So we have information about only highly expressed genes, at least in agreement between the cells. However, when we look on clones, you see that this plot it looks much better. Another, yes?
So these CloneSeek, in the sense that you grow cells into a clone, but afterwards it's single cell.
After what?
After that, it's single cell sequencing.
No. CloneSeek, it means that we grow them into clone, and then we treat each clone as a singular entity, and we sequence it as, we barcode as a bulk, as one clone.
So the barcode is for all the cells that are
In the clone. Yeah.
But then you do single cell from them.
No, yeah. I'm doing the single cell just as a comparison to try and convey that when we look on a single clone level, we have better information. Which makes sense because we look on more cells. the biological question, if it's meaningful, if it gives us to somehow studying the subpopulation underlying better or not. So this is the first clue we had that we are going to the right direction. So if we look on data coming from single cell and we ask what is the correlation between single cells, we get a very low correlation, which means that it might be a very nice biological heterogeneity. Most probably, it's related to the fact that we have very low sensitivity, very low complexity. But when we take cells and aggregate them, single cell profile, aggregate them together to form artificial clones, right? Then very rapidly we get, we achieve very high correlation. So we're actually dealing with bulk information. Like there is nothing interesting in comparison in the variation between 10 different profile of RNA-seq that were aggregated together with 10 other profile of single-cell RNA-seq aggregated together. But when we look on the clones and we separate them to small clones around 10 cells or above 10 cells, then we see that the correlation is lower, even though the number of cells that are being correlated here are very similar to what we see here. So that means, at least for us, that we have some information to extract, like variation information to extract from this type of experiment.
Are you sure there's no genetic differences between the clones? Like, it could easily be that there's copy number change in chromosomes.
Yeah, there are. And actually this is something that we would like to test, and it might be that this is partly the source of the variation. And now we currently, Shu in the lab is responsible for a full-length protocol adjustment to this, and then we will try also to look for some other mutations in future experiments. So then we just sequence the single cell or the clones, the equivalent Cloneseek. Again, the single cells are the same cells that came from the Cloneseek. We just separate them back into single cells. And I won't talk too much about the single cell because it looks boring. And again, PC9, nobody's doing single cell RNA-seq to them because they are very, very boring. And the one thing that you can see here that I colored is mainly cell cycle, derived by cell cycle. But when you look on the same data retrieved from the Cloneseek, you can clearly see some separation. And this separation, at least cluster 3 that marked here and 1, when you look here, they have a very distinct set of genes that upregulated them there, and they are all enriched for signature that are previously known to be related to cancer stem-like cells. Here we just look on the signature of cancer stem-like cells, and you can imagine a bimodal distribution here, even though it's a little bit hard because we are talking about relatively small amount of cells, of clones, but you don't see them on the single-cell RNA-seq. And what I forgot to show here is that this signature is composed from genes that are relatively lowly expressed. So it kind of explains why we could capture that with the clones, and we couldn't capture that with the single cells. Then we decided to move to embryonic stem cells, which is more of a complicated system. I just want to remind you that mouse embryonic stem cells has the core pluripotent regulators. I also added here manually the ESRB because we like it.
Question about the previous slide. I mean, it's an interesting result, but can you ascertain that this result to the developmental history of the cells within the hydrogel, or whether this is indeed just a matter of detectability of the transcripts because you're sequencing clones?
I don't have a very good answer for that currently, but I guess that when I'll finish the slide of the mouse embryonic stem cells, you might you might agree that
I mean, regardless of the answer, it's still it is
Yeah.
interesting.
So I will say before we get to the slide that it seems like the 3D condition promote pluripotency. So it might be that we also some kind of enriching the population of pluripotent like cells or something like that. So when we move to mouse embryonic stem cells, we have the ground state. And we have a state which is upregulated by UTF1, which are still pluripotent, but it's not exactly like the ground state. Both of them can later differentiate to all tissues of the embryo and the primitive endoderm, right? Or the XN, the primitive-like endoderm. So we have a cell line with three of these markers, these pluripotent markers, NANOG in green, UTF1 in red, and ESRB in blue. And what we did, we grow the clones in 3D, and we treat them with, and we culture them with 2I leaf, which is the common pluripotent supporting media. But we also try to culture them without 2IN leaf for 8 days. 8 days. And what we found is that when we culture them in the classical condition, we just get a very nice NANOG and ESRB and a little bit fader UTF1 because most of these cells reside within this state. However, after 8 days without 2I leaf, the NANOG is a little bit weaker, UTF is stronger, and ESRB is weaker, which means that most probably we have enrichment for clones that reside with this state. In comparison, the regular 2D condition, when we culture the cells on a plate, you see that the markers are a little bit dimmer, and after 8 days without 2I leaf, we see nothing. So the cells are randomly differentiated. And when we took those cells and cultured them back on fibroblast feeders, you can see that cells that have been growing without 2I leaf can go back to the ground state. However, cells that was growing on 2D couldn't, which means that our 3D culturing condition for mouse embryonic stem cells is really promoting pluripotency. And also when we did bulk RNA-seq and compared 3D versus 2D, you can see that most of the pluripotent factors are upregulated during the 3D culturing condition. Okay, so now we took the cells that we grow with and without 2I leaf in the 3D condition and differentiate them using retinoic acid for 4 days. And what you can see that there is a different potential to differentiate. The cells that were growing in this classical condition was as expected, as already published many times in the past, will enrich for neuroectoderm. However, the one that was growing for these days without 2I leaf were differentiate into endoderm. And the final part, the final thing in this part is that we try to do iPSCs, induced pluripotency, and also with induced pluripotency when we encapsulate a mouse embryonic fibroblast that has the OSKM cassette under the regulation of DOX, of TET, then around 60% of those fibroblasts could form very nice NANOG positive iPSCs. And the 2D condition is already reported in many other past work, is around 25% efficiency. So overall, our 3D condition culturing in hydrogels promote pluripotency. And finally, as I just told you, it's very hard to force those cells, those mouse embryonic stem cells within the hydrogel to differentiate because they prefer to stay on a pluripotent state. So in order to help them differentiate, what we did is we used a cell line in which controlled by a TET off of DOX for OCT4. So when we add DOX, we remove or downregulate OCT4. Then we treated the embryonic stem cells with 2I leaf and DOX for 1 day, and that's what you see here. And after that, we proceed with the DOX, but remove 2I leaf and produce those clones. After 4 days of random differentiation, because they lack both OCT4 and 2I leaf, we could get a very nice separation of the three main germ layers, endoderm, ectoderm, and mesoderm. However, when we break those clones into single cells, again, we got a kind of a blurred image. In terms there is some reminiscence of subpopulations here, but they are not really agreed with the same markers that I'm showing for the clones. And in order to, again, to explain this result, we looked for all the genes that are differentially expressed in the single cell assay and in the Cloneseek assay. We find a lot of genes that are intersected, but some of the genes that explain the separations are not really intersected. For the single cells are mainly highly expressed genes, metabolism, cell cycle, transport. For the Cloneseek differentially expressed genes, we found a lot of markers of differentiation, which means, and when we look on the level of expression, then we could actually detect them expressed in the Cloneseek. We couldn't detect them expressed in the single cell RNA-seq, which again suggests that the problem to detect differentially expressed genes that related to germ layer differentiation in single cell RNA-seq is because they are not really highly expressed.
And is it natural, as in you introduced this differentiation of Cloneseek, it is reflecting in vivo conditions? Because maybe you will promote this very tight hydrogel, other cells will promote the differentiation signal. Maybe some of them would like to remain pluripotent in the
You are totally correct, but you just forgot that the single cell RNA-seq is the same, are coming from the same cells exactly. We took the clones and we just break them into single cell. The only difference here is technical, right? This was profiled as clones, those profiled as single cells. We didn't culture them differently. And beside of that, I don't know. It might be. We know that in the hydrogels, differentiation is not something easy to get. And because of that, we needed to downregulate OCT4. Yeah?
Have you tried comparing Cloneseek with single cell RNA-seq when it comes to perturbation effects, so introducing CRISPR or something like that?
It's something that we are working on it. We still don't have it yet. Yeah?
Wouldn't it be better to identify the future genes using the clones and go back to the later single cells and use those future genes as
This is possible. The only problem with that, it might be that if we take these genes and try to apply them, so looking only on the matrix of these genes and trying to analyze the single cell RNA-seq, a lot of them will come back as zeros. So we couldn't even detect them on the single cell level. That's the main problem. If we could detect them, they already in the begin with will produce this nice separation that is really aligned with germ layer markers.
Have you tried doing a tooth map and seeing if they cluster at all, or is it something new method?
We did a lot of playing around with this. I don't remember if this is specifically something we did or not, but I can assure you that most of those genes are coming as zeros here in most of the cells.
Just try to compare it to commercial development single cell, like for example, like taking the same
Yeah.
clone, you broke them now, left them in single cells, and run them on the
We didn't try that, but theoretically it's supposed to give us very similar results. It's just annoying because it's too expensive. So
Oren, the experiment also suggests that each clone is only exploring one germ layer.
yeah.
Are the cells already primed to go into one of the three germ layers?
This is interesting questions. We are not sure yet exactly what happened there, but because I think that maybe it will align with the second part of the talk, and then you'll see what we try to understand is actually to study what happened in the exit from pluripotency. So it might be that those single cells already kind of prone to a specific germ layer before they make the clones, just because the OCT4 is downregulated.
So there is another method of using a single cell as a map and then compare like hundreds of boxes available to map back to your single cell. That's to increase the cell number also.
Again,
So there
we didn't try that. It's actually something that I remember now that one of the reviewers asked, actually, but we didn't do that. I think the main problem, we didn't really understand how it's supposed to work or something like that. But any, okay, that makes sense, that we didn't understand how it works, I guess. Okay, so what next? So now we're optimizing Cloneseek for primary tumors. The first glioblastoma I already showed you, but we're also trying brain metastasis of lung cancer now. We are developing full-length protocol to further improve coverage and mapping of somatic mutation and splicing events, which are currently not really detected on the single-cell level. And we modified the technology to allow profiling of chromatin from single clones. And when I say chromatin, I mean ChIP-seq and something that is equivalent to a TAC-seq. It's not really a TAC-seq, but it's something that allows us to extract enhancers. Okay, not sure how much time do I have. It was very, very slow. Okay, so the second project is more of biological oriented. Is been done by Sapir and Sharon in the lab. It's ESRB is a cell cycle dependent associated factor balancing pluripotency and XEN differentiation. XEN is the extra embryonic endoderm stem cells. It's something which is equivalent, the in vitro equivalent of primitive endoderm. When you take mouse embryonic stem cells, you treat them with retinoic acid, then you can get primitive endoderm and epiblast, the in vitro equivalent of those. So we found this nice paper from the labs of Van Oudenarden and Jaenisch, which done that in 2017 and actually took the stem cells, the mouse embryonic stem cells, treat them with retinoic acid, and you can see here the single cells. The mouse embryonic stem cells are very boring, as usual. And after 4 days of differentiation with retinoic acid, they got ectoderm-like and XEN-like. They also show that the exit from pluripotency happenings around 24 hours after retinoic acid was introduced. And after 4 days, they have this nice separation of XEN and ectoderm-like cells. So basically what they show is that exit from pluripotency using retinoic acid involves in the decisions to go towards epiblast, which is the embryo, and the hypoblast, which is the extra embryo tissues. But we are more interested to understand what happened here. Right? Here, it might be been argued that there is a stochastic effect, right, that makes those ES cells decide to go one direction and to the other. But for me, stochastic effect is mainly we still don't understand what's going on. Right? It might be that already here, and this is aligned with what you just asked, what happened here may be affecting the decisions. And the first option in mind that came up in our mind was cell cycle. And cell cycle, as I mentioned before, is a problem with single-cell RNA-seq because usually this is when you don't have something to find, you'll find cell cycle. So we decided to take this seriously and to produce a system that allows us to separate mouse embryonic stem cells according to their cell cycle states. We used both FTSE reporter and DNA dye, and eventually what we did is separated our stem cells into G1 and G2 phase. And this is, again, a limitation, technological limitation, because we can't really efficiently sort S phase, which is also very interesting. But G1 versus G2 is quite easy. So that's what we started with. And then after differentiation of cells that initiated differentiation from G1 or initiate differentiation for G2, we can profile and see what happened. So this is the first experiment, stem cells sorted, retinoic acid for 4 days, single-cell RNA-seq here, when they already mixed, at least according to the cell cycle. We don't care about the cell cycle at that stage currently. And what we found first is that, again, we could mimic some of the states instead of ectoderm, we call that cluster epiblast stem cell. And this was clearly XEN. And we also found another cluster which is mimic the mesoderm or the mesendoderm. There is a very strong GATA2 marker here. This cluster, we weren't sure what to do with it. It's mainly epiblast stem cell, but as you see here, it has better coverage, so we are not sure it's not technical. But most importantly, when we colored this map according to the cell cycle states those cells were residing before differentiation happened, then we found a very interesting phenomena that was repeated in many, many experiments, and is that the XEN cells were mainly derived from mouse embryonic stem cell reside in G2 state when retinoic acid introduced. So we start to ask ourselves what's going on there, why it happens. And we just did bulk RNA-seq to try and understand which are the genes that can explain this phenomena. Obviously, we found a mess. A lot of genes that come up as G2M, a lot of genes come up as G1, most of them related to cell cycle regulation, but not all of them. So we look in the literature of ourselves mainly, but not all. So this is a project we did with the Buganim lab. We're helping them. It's not our project. But anyway, one of the main issues here is that they found that when they try to make iPSCs using upregulation of ESRB instead of the OSKM, they actually produce a kind of a XEN state before IPS was completed. Okay, so we decided that ESRB is a very good candidate. Another project by Navarro showed that ESRB is actually a mitotic bookmarking factor, upregulating during G2. And we also found ESRB here upregulated during G2. So we decided to take our chances and put our money on ESRB. So first, we upregulated ESRB on G1 specific mouse embryonic stem cells. So we sorted G1 cells after upregulating ESRB in all the cells. And then we did again the differentiation, and suddenly we found XEN. This is the first experiment. Second one is knocking out ESRB. And again, after knocking out, suddenly we don't have XEN. So you see here, this is the epiblast. All of the knocked out cells were clustered in the epiblast subpopulation. You can see here some markers of XEN here, and you don't see the black dots. And then we wanted to understand the mechanism, but somehow, even though ESRB ChIP-seq is published, not for G1 versus G2M cells, that's what we wanted, so we tried to copy the protocol, but maybe, I don't know, 3 or 4 different ChIP-seq protocols. We couldn't make this ChIP-seq work in our hands, so what we did, we first compare K4ME2 and K4ME1 ChIP-seq for wild type and knockout cells. And we indeed found some enhancers that been down-regulated in knockouts. We also found that on wild type, the main distal enhancer that being captured by K4ME2 are related to developmental and differentiation. However, in the knockout, we have less enhancers that are associated with differentiation. This is, of course, not a good enough explanation for the specific XEN deprivation, but this is the best we could get. And when we took the ESRB ChIP-seq from a Navarro paper, so this is cells that are actually during interphase, so they're mainly S phase. And we found that ESRB is really associated nicely with enhancers that related to XEN. Finally, how much time do I have? So finally, what we try to do is to promote XEN direct differentiation. This is a 9 days protocol that eventually allow us to produce XEN In parallel, we tried to differentiate the cells into EBs. We found that ESRB knockout stem cells can form EBs quite nicely, equivalently to the EBs produced by wild-type cells. However, XEN could not be differentiate using ESRB knockouts. So mainly what happened is most of the cells are dying, and some of the cells, very small amount of cells, look really weird. We are not sure what they are even. But eventually, after day 9, you see a lot of XEN-like cells in the wild type and almost nothing in the ESRB knockout cells. So we came up with this hypothesis, still working on it, that in G1 we have lower amount of ESRB comparing to G2, and because this ESRB is associated with XEN-specific enhancers, then the chance of a cell that initiates its differentiation from G2M to choose the XEN program is higher comparing to cells which initiate their differentiation from G1. And G1 cells will prefer the epiblast, the embryo-like tissues. And now we are walking, so I just show you the mess that we see here. So ESRB was just a good gamble, but it might be that there are other genes that regulate differentiation on a cell cycle dependent manner, and we need to find them. We need to find the stories. So we decided to follow this finding with a CRISPR screen. So what we do, we have a guide for 20,000 different genes, all of the genes in the mouse genome, basically, times 4 guides for each gene. And after we find the essential genes for pluripotency, as they removed from the population of cells that we are growing, now we can sort the cells according to G1 and G2M, differentiate them, and ask what are the genes that are essential for differentiation from G1 cells, and what are the genes that are essential for differentiation from G2 genes. And this is very preliminary data. We just got the results. I'm showing it here. Don't spread it around. So this is the fold chains of the essential genes that are upregulated. So what is up? It's not upregulated. It's the proportion of cells within the population of differentiated cells. If we get a lot of reads that's supporting a specific guide, it means that it's not essential for differentiation. It's like the opposite of naive. Right? So if you have here a lot of genes that are associated with G1 because it's G1 versus G2, it means that they are essential for differentiation from G2. We didn't think about a better way to present it because it's very, very preliminary. I guess that we will need to find a better way to show it. But what we found here, for instance, is genes that related to PRC2. There are a lot of other genes, but I just mentioned here several genes that related to differentiation, transcription factors, SOX17, endoderm, HDAC1, FOXQ1, and EZH2. And you see here, JARID2 is one of the best scored here. So it's really, really, really needed for G2M cells to complete their differentiation. We have no clue why yet, but this is kind of the results we have. Okay, I think I'll finish with that. I would like to thank to the people that did the work, the lab members, our close collaborator Amnon Buxman, Eran Mashor here, and Alva Biran help us with the IPS. Yeah. Okay, I just finished. Yossi Buganim for close collaboration and the funding. Yeah. Sure. You need to speak louder.
A couple questions for the second part first. In one of your initial analysis, you looked at the G1 versus G2M bias for the differentiation. The single cell plot that you showed, I think it was showing for the G1 also majority of
Here.
the lower one. Sorry,
Yeah, this one. Yeah, it's here.
For the epiblast,
Yeah. So I just put here this heat map that kind of suggesting the proportions. I don't have the exact numbers, but yeah, this cluster is mainly enriched with G1 cell, but we didn't focus on this here because we are not sure what it means, because the first thing we got is that this cluster is composed from cells with better coverage. So we are not sure it's not some kind of technicalities. We have other replicates that showing actually similar results. So it might be something that is relevant, it's biologically relevant, but we are not sure.
And the focus part, this is also mostly green. Is
It's not mostly green. This one? No, it's misguided. It's kind of you see here it's cluster 1. You see here like it's more or less the same. It's just kind of hiding each other, hiding the red dots.
And the second question I have was about the CloneSeq. How many clones are you talking about when you get sequences?
We almost every time trying to get around 1,000 in every experiment. 1,000 cells, 1,000 clones. This is at some point we have a problem with the cost of the sequencing, right? And with 1,000 clones, it's already some kind of really exhaustive in terms of budget. Yeah.
I have a question regarding the CloneSeq. Have you investigated whether DPLX can go as clone?
It's something in my ears, I guess.
Have you investigated whether DPLX can go as clone with the CloneSeq and the DPLX?
To grow a clone? To? 2C? No, actually we didn't try that. To 2C state.
No, on DPLX cells.
Something.
Because from embryonic stem cells in ceramic conditions, we have a small population of DPLX cells. And in the field, it's like a big question whether they can actually revert and divide into more cells. So
Yeah.
if you had the clone in your CloneSeq coming from DPLX cells, it would be huge to know whether they can actually revert and go to more cells.
Yeah, I guess this can be interesting, but we didn't try that. We can talk about that later. We can plan maybe some experiment for that. It sounds like it's doable. We never looked at it.
Okay, Oren, so we have to finish. Let's thank Oren again. And we'll meet here at 10 past 11 sharp for the next talk. Thank you.
was very nice. Also CloneSeq. Just one question. For CloneSeq, if it's just
Starting up a little bit on how we are developing products in a company, if that sounds interesting to you. So at Diagenode over the years, we had the opportunity to develop numerous products around where is the okay, yes, good, around chromatin analysis, DNA methylation, lately non-coding RNA, and of course the fragmentation device that started the whole company in the beginning. And it doesn't really matter whether we are talking about instruments, equipments, kits, or antibodies. We always follow a similar type of product development model that I would like to talk to you about right now. And actually here it is. This is a fairly, as you can see, simple 5-step process from the very beginning of the development, named the scouting. Then we go through the pilot, the development, the validation, of course, and then the release when the product appear on the market. And you want something that is, of course, simple, so that it's easy to follow, reproducible, and that you can also apply to every type of developments. And with that, we can start with the first phase, so I will walk you through the whole 5 steps so you can see. Before I forget, I need to mention that I will use the non-coding RNA-seq, small RNA-seq developments I've been personally involved in as a support for this presentation. So you will see the development throughout this product. And the scouting, well, this is really the phase where we are trying to figure out first what we want to do. We don't go like that into the lab and start pipetting like crazy. No, we first need to understand a little bit where we want to go and what we want to do. So that's why we are using the phrase, we see what we release, then we develop, and not the other way around, like we develop, then we see what we release. And this can happen, this type of situation, and it's rather uncomfortable, I would say, because you end up with something in your hands and you don't know what to do with it. You don't have a market for it, no one is interested to buy it, so you basically failed before you even started. So the phase of the scouting, like I've said, is really the goal to understand needs and limitations also of the field, the market. And for that we are going through those steps. I mean, it's common sense, but it's quite important to cite it, I think, is that we first go through an extensive review of the publications that are already out there. We are also gathering a lot of different actors around the table, because I mean, those type of decisions are not made based on an RNA decision only. We have some sales and marketing also, we have management that is chiming in, and also at some point external collaborators, advisors. So it's really a multi-department decision process that is that is behind that. Something that is quite also important when you are working in a company is also to do some market research. So I think that's pretty unique, of course, to a company environment, is that you need to understand when where the competition is, what would be the future trends of the market, and try to understand a little bit better what are the solutions that are already out there before you start entering that field. And the last point that is quite important, I would say, is that also a company doesn't really stand on its own. I mean, you can take examples from all over the place, Diagenode and others. You don't have a company that is doing everything on its own. So it's always better when the company is embedded at some point into the field of research. And like I've said, I will use the small RNA-seq developments I've been involved in to show you a little bit how it was for us during that scouting phase. And for the small RNA-seq, I think it's important to first remember that a library preparation is a means to convert information from a biological sample to the sequencer. So the information has to be read by the sequencer, must be readable by the sequencer, that's what I mean. And the sequencer will release, of course, data that you can analyze and find some results to the question you have been asking in the beginning. But the process that are really going on between the sample and the sequencer are actually very critical to convert that information and to preserve that information that you have in the sample in the beginning. And when we are looking at, let's say, the industry-leading kits out there on the market that are doing small RNA-seq, we can easily say that they are always, or they are really, really often times based on RNA ligase, and by that design only, by that choice, I mean those kits will tend to preferentially enrich RNA that are 5' phosphorylated and 3' hydroxylated. So choosing the technology for the library preparation is actually very important, because this will impact your future results. So this, of course, this choice, there is nothing wrong about that, but you need to be aware of it, impacts, of course, the library content quite a lot. And as a result, you will typically see an enrichment in mature microRNA and at the expense, let's say, of the other non-coding RNAs that you might have in your sample in the beginning. And this is how it looks like, basically. So you have a whole bunch of non-coding RNAs that are floating around in the sample, and by applying this type of library preparation, you are selectively enriching a certain type of non-coding RNA. So I think choosing the right library prep kit is actually changing qualitatively and quantitatively the representation of the sample. And doing that during the scouting phase, we stumble upon this publication from 2014, published by a German group from the DPLX named Capture and Amplification by Tailing and Switching. And this workflow is actually completely different from everything we had the opportunity to see so far, because it doesn't use any RNA ligase. And in a nutshell, you can see that this RNA molecule is first 3' end poly-A tail by an RNA polymerase. That poly-A tail is used to prime the reverse transcription thanks to an oligo-DT primer, and this oligo-DT primer as a part of the Illumina adapter. And we can start then the cDNA synthesis, here again in blue, and choosing a special mutant of reverse transcriptase. During the cDNA synthesis, the reverse transcriptase is doing what we call the template switching. So this is really key to this technology. And the template switching consists actually of adding a bunch of a series of non-templated nucleotides at the end of the cDNA strand. And this addition of non-templated nucleotides is guided by a template switch oligo here in green with a complementary sequence. And recent papers, mainly from New England Biolabs, have shown that it's actually the template switch oligo that is guiding the template switching and not the other way around. And you can see that here CCC, GGG, it's simply because this is the most efficient motif for template switching.
For the poly, does it need any hydroxyl at the 3' end for the poly-A tailing?
No, we can do an end repair of the 3' end, so that this is a 3' end hydroxyl. Not based on the theory, I would say yes, but in our results we are seeing a lot of different non-coding RNAs that are not especially terminated by a 3' hydroxyl.
Because you can do the same end repair with RNA ligation, right?
Yeah, we can do that, but this RNA polymerase seems to be a little bit more flexible and versatile to incorporate more different types of RNA rather than typical RNA ligases. But it's true that those RNA ligases kits have evolved also quite a bit since the past years. And here we have, so after this reverse transcription / template switching, a P5 adapter, a P7 adapter, so it's really easy now to PCR amplify everything and get double-stranded DNA that can be read by the sequencer. And here, reading that paper, it was interesting for us to extract what we call in this type of product development, kit development, the proposition value. So what we are going to propose with that type of kit, with that technology. So for us,
just for my understanding, so how many patents, for example, at this stage do you have to look at? Because I guess template switching is patented, right? The polyadenylation is patented, right? So this already probably has a couple of patents already included.
yes. Well, we actually only took that one patent for that specific paper.
That paper had to then, I guess, reference other patents.
Well, yep, but I mean, it's apparently it's something we hadn't really to deal with. So.
But as a company, I guess the best is no patents set up, right? I guess that would be economically the most favorable.
No, not really, because well, I mean, it's not really the model we are using. In this particular case, that paper, we bought a license to use that technology, and this group is receiving a royalty for every kit we are selling, and so this is the model we are following, but yes, we actually, to kind of protect ourselves, decided to also patent with that group this particular technology, but it's actually very difficult to patent because I don't think the poly-A tailing is actually protected for RNA molecules, so that's it. And for the template switching, we have not been bothered by that too much, I would say. So
Your patents are for the device.
yeah, well, patenting, it's difficult and complicated, I would say. Based on those reactions, there is not a lot of things to patent. We have simply a small patent, if I believe it will come later in the presentation, to protect a little bit the way we design the template switch oligo. But I mean, as far as I know, patenting the RNA polymerization is not applicable in that case. And the template switching is, I don't know about that patent. That's a good question.
CloneSeq technology.
Yeah, it's CloneSeq in the beginning. Yeah, definitely. No, I have to say I'm not really well informed regarding the IP consideration here. But in this technology, what was important for us during that scouting phase when we are trying to figure out what we want to do is to have this proposition value, because well, honestly, the best way to have a kit or a technology working is when it's fitting a need, or when you are filling a gap, if you can insert yourself in some sort of niche, let's say. So making a copycat of something that already exists is not really interesting. Here, just the three pillars, let's say, of that proposition value is being able to recover a dense, comprehensive non-coding RNA data set, being able to work on low quantities of RNA, and of course having a protocol that is simple, one day, one tube, no breaks or no purification in betweens. So that closes the first part. Now we have a direction where we want to go into. We know we at least have an idea of what we want to do, and we can move on to the pilot now. So the pilot, this is actually making the project on paper, a plan of the project, actually before going into the lab and again start pipetting. So we first need to have a plan first that we can submit. And for that, in a company, there is always a sort of balance we need to respect with the so-called requirements of our future kit and the expenses, of course. So we need to find some sort of balance between the two. For that, it's also a type of multidisciplinary kind of decision with R&D management, sales and marketing, and everyone is involved in this type of stuff. And with the requirements, something that could be interesting is that we are, let's say, defining a domain of application in which we want this kit to work. So for that, we are using the must, should, could, and want. I will come to that in a minute. We are also trying to figure out what would be the technical specification of that kit. Of course, also what would be the success and failure criteria that we can track along the way during our development, so that we don't make any, let's say, we don't drift away. And this is balanced with all the big questions like how much money are we going to include into that? How many persons are going to work into it? For how long? Do you need to purchase any equipment? Do you need any training? And that type of stuff. So it's always the risk-reward ratio. And for that particular technology, I would say the main interesting domain of application for us has been to take it and rebrand it, so to speak. Now it's called the DPLX or DPLX, DPLX, for cell-free RNA application. And we define that domain of application like that. So for us, this technology must work on cell-free RNA sample first, of course, for as low as 200 microliters of plasma. It should be able to capture the non-coding transcriptome, definitely. And it should be easily automatable, user-friendly, and we also decided to include a means to limit the PCR amplification bias. So the must, should are the requirements. Without them, we cannot launch. The project is stalled. So we must reach them in order to have a release. And then there is the could and the want. That would be nice, would be optional, would make the kit even better, I would say, with this, for instance, having some specific small non-coding RNA enrichment module. That can happen, that can be done. And also, quite important, defining the want. It's as important, in my opinion, to define what your kit can do and what it won't do. And in this case, we decided, well, this is not, we won't go after, we won't compete with the microRNA-seq kits based on RNA ligation, because we are simply proposing two different type of tool for two different type of questions. And we typically end up, let's say, the pilot phase with a big mind, that worked, with a big mind map, like here it's condensed for the sake of the presentation, but it's a mind map where we try to list, let's say, every experiments we could do in order to meet our objectives in the beginning. So, for instance, here a big cluster of experiments for us was to decrease the background noise, because that technology was prone to generate oligos interaction, dimers, co-products as we call them. So we wanted to make sure that we get a clean signal without any contaminating co-products, especially given the fact that we are working on low input. Also having a clusters of experiment like here, where we increase the incorporation of RNA as much as possible in the library, because that's what we want in this case, is having a comprehensive data set so that we can analyze a lot of things at the same time. And also clusters of experiments that were considered for small non-coding RNA enrichment, depletion of abundant transcripts that can be done too, and size selection of on-target fragments. So now we've cleared, let's say, the first two steps, the scouting, the pilot, so we have a pretty clear idea of what we want to do, and it's time for now to do it with the development. So now it's time to go into the lab and start making experiments. And with those, the development typically moves from one experiment to another one, and we get results, and at some point, if we have something that is good enough, we decide to go for a release. But something that I wanted to mention for the development here is that it's important for us to stay on target as much as possible. So that's why whenever you have a team of researchers constituted to work on that project, we don't leave them alone, so to speak. We have some frequent checkpoints, we have some other researchers from the company that can check the results and the progress with them. We are also oftentimes asking collaborators, external partners to come and have a look at our project and what we are doing, and this is quite important to us, to stay open-minded and avoid some tunnel vision and fixation. So I think that's quite important to mention that in the development. And another thing, whenever you are leading, let's say, a team of researchers to take care of the development of a project, those questions are popping up. And I wanted just to highlight the fact that those questions are as important to me as the technical developments. Having a team of researchers running smoothly is as important as making experiments. So that's why you need to sort out how do we all communicate, how do we record the results in a similar fashion, everyone following the same recording system, and how do we share the workload among the different researchers inside that team. So here I don't mean to bore you to death with those experiments that we did, but I mean just to give you an example of what we decided to change from the original version of the paper to what we have in a kit right now, and to fit our objectives. And for that, we decided, first of all, to include a UMI tag in the TSO, to change the TSO sequence and modify it so it won't produce co-products as much as before. Include a crowding agent in the protocol so that we can increase the incorporation of the RNA into the library, because we really see it as a means to convert information, so we don't want to lose any information here. And also, it basically boils down to breaking up a kit apart, a protocol apart, and reassemble it in the best way possible with optimized condition reactions, like if we can find additive enhancers that would make an RNA polymerase work better, or a reverse transcriptase, or a DNA polymerase for the PCR amplification. That's why I think we are a little bit more flexible in terms of RNA incorporation than other technologies. So you can see that this is very technically oriented type of experiments. We are really looking at details in a protocol. I thought it might be interesting to mention. And now, regarding the validation, so the development has moved from experiment to experiment. Typically, it can last from, I would say, 6 months to 9 months of experiments, and sometimes 4 people working on it, so this can be quite extensive. And at some point, let's say that we will decide that we have something that is good enough for a release, so we decide to validate, of course. So during that validation, I like to take the analogy, let's say, with an IKEA furniture, because a kit is really like an IKEA furniture, I think, because you want it to work different times, different places with different users. Otherwise, I don't think this can be called a kit. That's what is, I would say, making the biggest difference in a company, is to make sure that whatever you have developed will work anytime, anywhere, with different users. At least we can try to make that work. And for that, we typically designed a validation panel, or sometimes multiple validation panel, to reflect that intended use of the kit, that domain of application we were talking about, to make sure that this technology, this protocol, will handle itself quite well in the different stresses we make it run through. And this is the time where we are, of course, defining all those technical characteristics that must be defined at that point. So what is the sensitivity limit of the method? What is the robustness of the method, and how do you challenge it with what type of sample, different template preps? Something that is quite important also to us, like I mentioned, is the reproducibility. Make sure it works across different places, lots of regions. We already had the joke of having something that works, and then you switch and you change and you take another lot of regions, and that doesn't work anymore. So that's bad. But you need to make sure that the reproducibility is on point, and of course you fix all that those performance, I would say, of that future kit that you are about to release. And once this is done, once you are confident, let's say, I almost forgot, here regarding this DPLX on cell-free RNA, a couple of results that I could show you that are validating, so to speak, the domain of application. Here I'm showing just the library content at CPM 0, so it's of course a very low detection threshold, but you can see every transcript that are captured on 100 picogram of cell-free RNA, so it's actually less than 200 microliter of plasma most of the time. And you see that we are detecting a lot of protein coding genes, in fact. We are detecting a fair share also of long non-coding RNA, small non-coding RNAs, here pseudogenes, and a little bit of tRNA and so on. And typically, if you decide to raise up the CPM threshold, you will see that you will start losing a lot of this long non-coding RNA and protein coding signal, meaning that it's seems that we detect a lot of those, but at a very low expression level, products of degradation certainly. And the small non-coding RNAs here, this is just a zoom of the different small non-coding RNAs that are encaptured in the library with a very big proportion of piRNA, miscellaneous RNA, microRNA, SNO RNA, and also snRNA. So typically this type of data set is quite complex, and we have a lot of data to interrogate for the future. Like I mentioned, in this particular kit, it was important for us also to correct the PCR amplification, because we are working on very low amounts of RNA, very low input. So from 1 nanogram typically down to 10 picogram. We had researchers using this kit to analyze the non-coding transcriptome of a dried blood spot. So it's a very low quantity of RNA. And here it's just to say the fact that when you are taking into account UMI deduplication, after UMI deduplication and here before, you have the black line that is showing you no difference between the two. And as you go down in input, you see this cloud of dots that are small non-coding RNAs shifting away from the black line towards before UMI deduplication, just meaning that there is as you go down in input, there is an increasing importance to take into account the PCR clones that are arising in your data. Otherwise, you may tend to overestimate the expression of your transcripts. And finally, something we also wanted to make sure is to compare with RT-qPCR and see how this was holding up. So this is the NGS fold change over the RT-qPCR fold change for those 20 small non-coding RNAs. Everything has been relatively expressed down to SNORD49A that we see here, and you can see that those changes in expression for the NGS data and the RT-qPCR data are more or less consistent with let's say an okay correlation between the two. So this opens up also the door for semi-quantitative assessment, let's say, of the small non-coding RNAs. And finally, something that quite important for us is also to make sure that a data set in terms of transcript detected is consistent across input. That may sound like stupid, but it's actually very important that your kit will produce the same results every time across the whole input range that you decided to validate. So those are just a couple of validation results that I thought would be interesting to mention. And now it's time for the release. So this is the moment where yes, sorry, go ahead.
Do you offer some kind of quality control pipeline for the analysis? Or are you going to use the kit?
In terms of data analysis?
Yeah.
Yeah, I will come to that in a minute. But yeah, it's actually a need we felt like fulfilling a little bit. Like we saw during our development and from feedbacks from collaborators and customers that analyzing non-coding RNAs can be challenging at some point, and we decided also to invest in bioinformatical development and propose a tool that would help resolve the ambiguity there. And I will just mention that during the release, and actually this is the release, so things are getting a little bit crazy at that point usually. And it's quite interesting because now you've spent typically, let's say, 9 months in the lab working on a protocol, so you are a little bit sick of seeing always the same bioanalyzer traces in terms of library preparation, and by that time you know the protocol by heart and you dream about it. But now you have the chance to interact, let's say, with the other departments of the company, because at least at Diagenode, I wouldn't promise you that it goes like that in the other companies, but it's the R&D who's working on the user manual, so how you are translating your work into something that can be used by anyone who wishes to purchase that kit. You also establish a QC procedure that will be followed by the production department to make sure that every production they will make will be good to go and ready for sale. You are also like transferring everything needed to the production department to make sure that they can produce your kit correctly. You are designing a web page, so if you go look at the Diagenode web page, you can see all the products that we have, and typically we are involved in this design as well. And importantly as well is training the sales team that will go out there and try to sell the kit for you at least. And but in terms of R&D, something that I needed to mention is the fact that the adventure is only beginning at that point. We have the 5 steps, so we can go to the release. The kit is perhaps technically working, but you don't know yet if your kit will work in the hands of, let's say, real researchers in real application. So that's why it's really important to start collecting feedback from the field. So typically we can run through, for instance, a beta testing campaign to see a little bit how the kit handles itself in other situation. You get a lot of, let's say, feedback through our tech support channel, and if you are lucky, at some point your kit will be used in scientific publication, and that's always, let's say, gratifying to see that this technology has been applied to make, let's say, real science, and at least to make progresses in that vein. And here I just wanted to mention the fact that this is actually coherent with the domain of application we had in the beginning. So we developed a tool for non-coding RNA, low input, biofluid types of sample, and this is coherent with, let's say, what we see in the publication when the kit is used. So that's always nice to see, of course. And to finish, I just want to leave you with this, loop of improvement, this feedback loop, because the feedback we are receiving are always driving changes and improvements to our technology and protocol. That's why you are seeing, for instance, a V2, V3, V4 kit. It's always an ongoing process, I mean. And this feedback loop, in our particular case, like I was just mentioning, we decided to address the fact of non-coding RNA alignment and multi-overlapping and multi-mapping ambiguity by that tool called MGCount that was actually written and developed by one of our PhD students. And this tool has been recently published. It's free of use, and it helps, well, at least it aims to resolve the ambiguity in non-coding RNA data analysis. So this is a type of example, let's say, that can come out of this feedback loop. And it's already time for me to wrap up. This was a short presentation, so we can have the chance also to exchange in a live Q&A if you wish. And to finish, I would say that following a systematic way of developing a product ensures us, first of all, a consistency across the whole R&D department to make sure that every team of researchers is working in a similar fashion. To make sure that we also minimize the risk of mistargeting, meaning that we hit the wrong target and we have a kit that's not going to be successful. And always following a system, I would argue that it's always beneficial to cut down in terms of delays and missteps along the way. And ultimately, I would say that increases the technical and ultimately the commercial success of that project. And with that, thank you for your attention.
Okay, well, thank you very much, Gilles. Do we have questions?
So for statistical reasons, when you start this process, how many make it to the end?
Yeah, in terms of project, yeah. Not 100%, definitely. Sometimes they are dropped during the development, sometimes they are dropped during the validation. It's not it's a system. I mean, for now, it's the best way we have to develop products, but it's not ensuring us a 100% success rate of everything that we start in the scouting. Not everything will see the light of the day of the release.
Is it closer to 90% or closer to 10%?
Yeah. In that range, I would say no. In my experience, so I've been working for Diagenode for 7 years now, and for several reasons. Sometimes it's just because it's bad positioning and we have something that's not working and that's not commercially successful, or sometimes it's just because the development is not on point and we made a mistake. I would say that in 7 years, I know three, 4 failures of projects that didn't work at all. In perhaps I would say a dozen maybe of projects. So it can be, of course, a lot of different reasons, but yes, it's not a perfect system at all. Yeah. And I think every company is going through that at some point or so. I mean,
normally in the lab, it's like 5% of the project really works.
Yeah. But that's why, I mean, that's why in this case, I would argue why this would work a little bit better. I mean, in terms of commercial development, of course, is that we first try to understand as clearly as possible, that was the point I was trying to make in the presentation, what we want to release to make sure we are fulfilling, let's say, a need or a gap or something like that. It's the best guarantee we have that it can work. Beside the fact that, of course, the development can fail at some point. But, yeah.
Questions? I'll ask one. So, who decides on killing the product?
Yeah, good question. I mean, it's a mutual decision, I would say. It's not always from the part of the R&D, if you ask my personal opinion, because when you are invested in a project, you don't necessarily want to drop it, but it's always about that balance of cost and reward and risk ratio and the thing I showed in the beginning in the pilot. So with that, I would say it's the final decision comes down to the management at some point. Yeah.
No breakup is really mutual. Yeah. Other questions? Yeah.
How you have an R&D, probably you have more than one project.
Yes.
Probably more than several scientists working at the same time.
Yes.
In the same project and in other projects.
Yep.
How do you make sure that everybody is not finding the same thing, but rather that what one finds, the other people can
Yes.
use it?
Yeah.
Time fashion, like in the
Yeah, I see what you mean, and it's actually a good question. We don't really have teams competing with each other to see who's gonna get to the end first. It's done in other companies, but it's not a strategy we really follow at Diagenode. We decided really to break down the R&D force, so to speak, into teams for DNA methylation, chromatin, non-coding RNA, instruments. And inside that team, yes, typically we are handling 2-3 projects at the same time. And this is this team that is responsible, let's say, for the non-coding RNA, small RNA, and another team is responsible for chromatin. And that's why during the development, it's quite interesting to us to stop at some point, have a checkpoint, and come and ask to the other chromatin researchers, DNA methylation researchers, can you have a look at my project and see if there is something I don't understand or miss. So that's why we have a lot of dynamic and communication between the sub-team inside the R&D department. Yeah.
Other questions? Okay, let's thank Gil and all the other speakers for and before we're leaving for lunch, Oren has an announcement.
So again, I want to thank everybody, and maybe just to emphasize that Episystem is really, research, and we have a collaboration with the industry. So it's nice to see this side of things.